Thank you. Good morning and welcome back. Today is the fourth day. Put the mic a little bit down. Yeah. So today is the fourth day. Welcome back. We have 4 more lectures. Today we have 2 and tomorrow we will have 2 and then we will be done. Today we are going to focus, in today's lecture we are going to focus on models, both in vitro and then in vivo models. What are the models we are going to use in the lab studying the mechanism of developing hypertrophic cardiomyopathy, how a single gene mutation translate into disease. So again in humans, in the last 3 days we talked about all the clinical conditions, etc. But when patients, they come to the hospital when they have symptoms, it's already been developed, or if you know already the proband, then you do the genetic screening in young their siblings or the children, and then you can expect down the road, but you have to do every 3 years, 2 years echocardiogram or ECG, everything. So, so we have to define a genetic variant, whether it's really a pathogenic or non-pathogenic, etc. If it is pathogenic, how it works, so that we can understand the disease mechanism and then we can treat the patient. So it's kind of understanding the disease mechanism. So what we are going to look in today's lecture is what are the role of in vitro experiments in hypertrophic cardiomyopathy research, and how these in vitro experiments will help us to dissect exactly the mechanism, and then how in vitro experiment can be complement before or to do the in vivo animal experiment or before going to the human studies. So again, these are in vitro experiments are the first step, and the cost, the investment compared to animal study much more less, and also time consumption is also less compared to in vivo, where if you look into animal model, you take time to breed, maintain the line, and so on. So in vitro experiment, for example, protein-protein interaction studies, you can generate recombinant proteins, and then you can really see whether the mutant protein interact with the other protein compared to the wild type protein. So it's all like you can make quick understanding of the disease. So going back, so there are several types of cardiomyopathies based on the phenotype. Hypertrophic cardiomyopathy, dilated cardiomyopathy, you have the ventricles very enlarged, the walls are very thin, and then restrictive cardiomyopathy, where you have stiffened left ventricular walls, and then irregular arrhythmogenic cardiomyopathy, and then non-compaction cardiomyopathy. So there are various types, but again we are focusing on hypertrophic cardiomyopathy in this course of work. Also, we studied that hypertrophic cardiomyopathy is the disease of the sarcomere, because mutations are predominantly in the sarcomeric genes, starting from myosin heavy chain, myosin binding protein C, and rest of the thin and thick filament proteins. So the first model currently being very well used is the iPSC derived cardiomyocytes. So as you know, in Japan, Dr. Yamanaka discovered how to convert a fibroblast into stem cells through induced pluripotent that cell type. So now we can use from the WBC cells, urinary progenitor cells, fibroblast, endothelial cells, any cells you can convert them into iPSC. From the iPSC you can then convert any type of cells. So here we are using the iPSC derived cardiomyocytes, Again, the conversion or the differentiation is a very specific protocol for each cell type. So, if you want to convert iPSC into myotubes or skeletal muscle, you use different medium or the differentiation factors. Whereas, if you want to convert iPSC to cardiomyocytes, you also use differentiation medium with the differential factors. So, based on what cell type you want to generate from the iPSC, you have to use the differential mediums. So, again you can collect blood sample or skin biopsies. From that you can convert them into iPSC. Once you have iPSC, you can generate a lot of cells, you can freeze them in liquid nitrogen, you can take one aliquot, convert into cardiomyocytes, and then you can study the property. So, I am going to walk through today some of the examples from our own lab as well as from other labs. So, again the advantage is you can collect the samples from patients. So, you can have patient specific iPSC and derived cardiomyocytes. Sometimes we also do the isogenic iPSC derived cardiomyocytes. You take a normal cell line and then you introduce the mutation by using CRISPR-Cas9, and then you look for the phenotype in the cardiomyocytes. So, you can compare the patient's derived cardiomyocytes versus isogenic derived cardiomyocytes, confirm that the mutation is truly contributing to the contractile dysfunction and then the disease. You can also correct the mutation in the iPSC in vitro and then see whether you can rescue the phenotype. So, that is another advantage in vitro studies. You can test various molecules, various compounds, whether you can improve or rescue the phenotype. So, the advantage is it is a directly human iPSC or human model, but again there are a lot of disadvantages using iPSC derived cardiomyocytes. So, we can do large scale, multi cells, multi wells, high throughput, we can do a lot of things. And then we have the isogenic controls we can generate. So, what are the applications of using iPSC derived cardiomyocytes? Number one, it is a sarcomere disarray. So, again if you want to know whether a mutation can cause sarcomere dysfunction structurally, we can look into these iPSC derived cardiomyocytes under electron microscope, and then you can see them. Calcium handling, for example, there are a lot of techniques can be used to measure the calcium handling within the cardiomyocytes of these iPSC derived cardiomyocytes. Again, some mutations, for example myosin heavy chain mutation, you can look compared to troponin I, troponin T, troponin C, the calcium handling will be very much different because they are thin filament protein. If you modify by any mutation, the calcium binding affinity can be changed. So, that would alter calcium handling in the cardiomyocytes. Electrophysiological abnormalities can be studied, and then drug discovery screening. For example, mavacamten was discovered using such a in vitro experimental conditions. And then last, recently we are doing all the gene therapy and genome editing within the Petri plate in the in vitro experiments. So, a lot of advantages, and the last few years ago in the United States FDA approved using the in vitro iPSC derived cardiomyocytes as a model. If you have drug screening, you can take the result, you can justify for a clinical trial. So, before even you go to animal model, you can start the foundation. So, again there are a lot of limitations. There is no perfect model we have. We have to really know what are the limitations of using the iPSC derived cardiomyocytes. Number one, the immature phenotype. What does that mean? So again, adult hypertrophic cardiomyopathy, as you know, mostly it is in adult stage, whereas the iPSC derived cardiomyocytes are mostly like embryonic stage, are having a lot of embryonic genes expressed in the iPSC derived cardiomyocytes, because they don't have the 3D structure, they don't have all the developmental stage. So the cell maturity is still not closer to an adult cardiomyocytes. So that's a big disadvantage. But other than that, so limited contractile force, again, as it's an embryonic kind of mimicking cell line, it cannot be directly compared to an adult cardiomyocytes. So right now, a lot of laboratories, they are working on how can we improve the protocol, taking this iPSC derived cardiomyocytes and make them as an adult, real adult matured cardiomyocytes. So with these limitations, but again, now the 3D model has come in, and today I am going to talk about the cardiac organoids. They are like a spherical structure and much more better than the 2D iPSC derived cardiomyocytes. And again, in this model, this is what I want to bring to your attention. A lot of bioengineering students get involved in this project because they can develop tools in such a way that when you plate the cells in a petri dish or in a culture condition, how can you give the kind of stimulation to improve the embryonic stage to adult stage, mechanical force, and so a lot of students get involved in this kind of project. The second, predominant in vitro experiments we do in the lab to focus on hypertrophic cardiomyopathy is protein-protein interaction studies. This is very important, right? When you have a mutation in a gene in a protein, you wanted to know whether it acts as a wild type or it modifies the protein structure in such a way that it either gain of interaction with any other proteins or loses its interaction with non-interacting partners. How do you do that? So again, you can do multiple way. The co-immunoprecipitation, where you throw the mutant protein or the wild type protein into the total lysate, and then you pull down with a specific antibody, and then you run in an electrophoresis gel. So you will see all proteins, whatever the proteins that interact or loses its interaction, you will see, and then you can do mass spec identifying all those proteins. And then the yeast to hybrid system is another one where you can have the non-interacting partners, whether if you already know in the literature in the interacting region, you can clone those regions and then you can see them in the yeast to hybrid system. If there is an interaction, then you will see color selection, etc. And so this is another way of doing it. The last one, this is the recent one, very much a lot of laboratories they are using it now. It's a real-time dynamic monitoring of binding. So they use biosensor, the labeling in the protein, both the partnering as well as the mutant or the wild type of your interest, and then see when they come together. You can do it in vitro in cells. If they come together, interact together, you can see the lights or the sensitivity, the signals. From that you can measure whether they are really changing their interaction or they maintain the interaction. Then other experiments are biochemical assays. Again, biochemical assays are predominantly to look for enzyme activity. You know that myosin heavy chain is an ATPase enzyme, and it can, based on the mutation, you can determine the ATPase, the rate of hydrolysis of ATP. So that can be simply done by an ELISA assay. You can isolate total myofibril preparations from the mouse heart or from the iPSC derived cardiomyocytes, and then you treat them with ATP and magnesium, etc., to activate the enzyme, and then see the time and then how much they can hydrolysis. Also, we look for phosphorylation status. Sometimes the mutations can alter the phosphorylation status, resulting that could cause the phenotype. And then other standard assays are binding affinity assays. So again, you can have 2 recombinant protein. It's kind of ELISA assay. You can see whether there is a binding or not based on the affinity. The Western blot mass specs, again standard. You can look for the expression level. For example, haploinsufficiency. When we talk about it, you can measure how much total protein of interest is expressed in the presence of the mutation. Again, you can look at the various developmental stage and then see at what time point the haploinsufficiency is happening. Western blot is very cost efficiency, but mass spec is a little bit more, but more accuracy. Again, based on how much money you have, you can do this experiment. But at the end of the day, the goal for all these in vitro experiments or approaches is to integrate what we know about the mutation, and then go from there. For example, if you have a mutation in myosin heavy chain, if that is increase in its activity, and we know it's a hypercontraction, and then it can be recommended to treat with mavacamten. Mavacamten is an myosin inhibitor. Tomorrow's talk we are going to talk about all the drug screening and available ongoing treatment options, etc. But mavacamten is a known clinically approved, FDA approved drug that been used in the clinic. So overall, the in vitro approaches really can help us to identify the mechanism and then propose drug screening or identify targets. From that, you can select, for example, from thousands of drugs, you can narrow down to 10 or 20, and then you can take them to do the next set of experiment. So again, with this with the in vitro experiments, definitely there are a lot of opportunities for translational aspect, from drug discovery point, from gene therapy, gene editing, all those therapeutic aspect, we can really make it to translational point using all these in vitro approaches. So in the next couple of slides, I am going to give some examples on how in our lab using these in vitro models and test our hypotheses, all the challenges we have in the lab, etc. So that will give you some idea on the in vitro experiments. Again, before I go into the real example, I want to see whether you have any questions or not. So again, we can open up for 5 minutes questions, discuss about your knowledge, what you know about in vitro experiments, have you done anything in the lab? If you want to share anything, happy to hear that. So anybody does anything related to the any in vitro experiments in the, yes? I think 1 second. Okay. 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 So you have some knowledge. Okay.
And then I have worked also with 3D organoid as well.
3 organoids,
Yeah,
okay.
so we have done development of genetically modified human intestine for study of enterovirus.
Okay, okay, that's very good. Again, it looks easy, but again, you have to spend time establishing the protocol, you know, every time, like, be consistent in the experiment, you know, so take some time to have it in the lab. Any questions on in vitro experiments? Again, each experiment is a separate lecture. You can spend on one topic, whole hour, just talk about how you do it. So that's a different topic. But again, at least we want to give you an overview of all the in vitro experiments we do in the lab. Also, at the end of the lecture, I will share with you all the literatures. I have over 200 articles being collected in the last 20 years. I have them in a folder. I will share with you all. So you can download from the link. Any questions? Okay, so I want to give you some examples how we do in the lab, both in vitro as well as in vivo experiments. So I told yesterday that we identified a 25 base pair deletion in myosin binding protein C, which is in intronic region, and that could cause potentially exon skipping or alter splicing of exon 33, as resulting that you modify the C10 domain of the myosin binding protein C. So this is an important region for the protein to be localized. So in the work we have done in India, we have 2 papers. One is JMCC, another one is Nature Genetic. Again, it's a kind of correlation study where you know, there are subjects carrying the variant, show the phenotype, but predominantly 95% of the subject are they don't have any phenotype. So we try to understand you know, whether the 25 base pair deletion is a real pathogenic or not. So again, I showed you yesterday that we collected 2,401 subject, identified all this heterozygous and homozygous. So what we did was we invited these subjects to come to the hospital where we were able to do echocardiogram. So these subjects were random, very random, in a public domain. We randomly went to the street, collected saliva sample, and then we screened for the variant. We have no idea what the clinical phenotype in these subjects have. So we invited them, we called them to come to the hospital. Again, it's not easy to bring somebody into the hospital. First of all, they wonder why, you know, oh, I give you the sample, why you are calling me, do I have a problem or not, you know. So we say that we have to say nothing, you know, all we have to say, we don't know whether, you know, we cannot even disclose whether they have a genetic problem or not. So we have to say, you know, completely unbiased, you know. So they come to the hospital, and then with our cardiologist, and they perform the standard M-mode echocardiogram to look for any clinical sign whether these genetic carriers have any phenotype. So in this study, we invited 35 non-carriers of the 25 base pair deletion, and then 47 heterozygous, actually 43 heterozygous and then 4 homozygous subjects we invited to the hospital. We were able to echocardiogram. Again, you can see the age almost we tried to maintain between the non-carrier and then the 25 base pair deletion carrier. So from this we can conclude if you have the variant, what is the outcome, you know, at least this data is enough to say that. So here are our echocardiogram data. So the left ventricular fractional shortening, which is contractile function, you can see there is a significant increase, but the level is 0.02, and rest of the echo parameters are completely normal, not normal. There is no difference between non-carrier and the 25 base pair deletion carrier. So there is a subset of subject, they are very severe, or they have shown the increased the cardiac function, but the rest of them are almost like very normal to compared to non-carrier. So the next question was, okay, what maybe this subject, they may have additional risk factors, and that is why they show the phenotype. So what we did was we performed the next generation sequencing, the true cardiomyocyte sequencing. It's a 174 gene panel. These are the list of genes we included in the sequencing. You can see there the genes and their association with various types of cardiovascular diseases. So again, our goal was to identify why some subjects have the phenotype and 95% are not having the phenotype. So we collaborated with the expert in genetic and cardiomyopathy, Dr. Beth McNally and Dr. Megan Roy Puckelwatch. They are at Northwestern in Chicago. So in our genetic screening, we identified another mutation, a missense mutation, aspartic acid to valine in exon 12, and this is a region that binds to myosin heavy chain to regulate myosin heavy chain function. So this was very interesting to us, and again there were like 12 subjects of the 25 base pair deletion carrier, they had this second novel gene variant. Not all of the 25 base pair deletion carriers carry this, and in fact this mutation is in the same allele with the 25 base pair deletion. So only 12 subjects were identified of the number of subjects we screened. So again the prevalence, when you say from the 4 million, 300,000 people, 25 base pair deletion, whereas the 10% of the 6% carry the second novel variant. So it's kind of within 25 base pair deletion, there is a second novel gene variant. Again, this comes from only Indian origin. So when we recalculated the echo function, you can see now the non-carrier 25 base pair deletion carrier, and then here we have 25 base pair deletion plus 389V. There is a clear difference in increased in cardiac function. So having 389V is definitely a pathogenic variant comparing to the 25 base pair deletion. So we do see that it's consistent in other cardiac parameters. So then we collected the blood sample from this subject and then generated iPSC derived cardiomyocytes. These are the cardiomyocytes stained with alpha actinin and nucleus. You can measure the cell size of the iPSC derived cardiomyocytes. Again, you can see compared to the controls having 389V, there is a significant increase in cell size. And then also you can also measure their calcium handling, because these cells are live cells, they beat. So you can really measure their contractile force as well as irregular calcium transients. So having the 389V, there is a significant increase in irregular calcium handling in the cardiomyocytes. So again, I talked about early on the limitations of having 2D iPSC derived cardiomyocytes. They are simply 2D and they don't have any other cells in the 2D. You are just focusing on cardiomyocytes. But in reality, in human or in any whole organ level, it's not just only cardiomyocytes. You also have fibroblasts, you also have endothelial cells, and then you also have immune cells, etc. So you don't have the crosstalk in the 2D iPSC derived cardiomyocytes. So that's number one. Number two, cell maturity is relatively moderate. It's not equivalent to an adult cardiomyocytes. And then if you look at all the cells in the Petri plate, the iPSC derived cardiomyocytes, there is a huge heterogeneity. You know, some cells don't beat like other cells. So there are some heterogeneity. So you have to be 100% unbiased when you do this experiment. So literature talk about all the limitations of the 2D iPSC derived cardiomyocytes. So the question is, okay, what is the next generation of in vitro model system? So obviously organoids, cardiac organoids, specifically iPSC derived cardiac organoids. So you can culture them from iPSC to spherical structure into 3D, and then you can have a matured organoid structure. So in the matured 3D organoid structure, you can have not just a cardiomyocytes, but also you can introduce any other cell types. You can also introduce blood vessels. You can almost like create like a real organ, but again it's a lot of you want to establish the protocol so well. So this is the next big thing for now in the research experiments. So a lot of advantages of using cardiac organoids. Number one, it's a 3D, so that's a big advantage. And then number two, they are relatively mature compared to 2D iPSC derived cardiomyocytes. Then the crosstalk between cells, so you can also introduce endothelial, fibroblast, anything else, then you can have much more real, I mean, when you look into the contractile force generation, if you have other cell type, they can also influence the function. So having this 3D model is much more better than the 2D iPSC derived cardiomyocytes. So before I go into that, we also performed in the lab, as I talk about protein-protein interaction. So this is Rohit Singh. He was my former postdoctoral fellow. He is right now at Amgen as a scientist. What he did was he generated a recombinant protein of myosin binding protein C, the N-terminal region from C0 to C2 with the 389V mutation or the without one, the wild type. So you have two different recombinant protein. You can do it in bacteria. You can clone this gene, overexpress in PET expression system, and then you can purify them from bacteria. And then we have the myosin S2 protein, because this is the region that interact with myosin S2. So the question we asked in this experiment is, having the 389V variant, will that affect its interaction with myosin S2? So the first experiment is the co-sedimentation assay. Again, it's a very standard. You mix two proteins, either the wild type and the S2, or the mutant and the S2. You mix them and then you centrifuge, ultra speed, and then you collect the pellet. And if there is a binding, and it will come in the pellet. If there is no binding, it will be in the supernatant. So when you run a gel, you can easily identify there is a binding or non-binding. So from that you can increase the concentration of the C0, C2, and then you can see the binding affinity. The second assay is a solid phase binding assay. This one is almost like an ELISA assay. So how you do it in a ELISA plate? You first coat the S2 protein, and then wash with buffer, and then you add either wild type or the mutant, and then see if there is an interaction, they will bind with the S2, and then you wash again, and then you add antibodies against this C0 domain, and then you can measure the signal in the ELISA plate. So that also will show you. And then here we do in vitro motility assay. This is a very unique assay where you have a glass plate, you coat myosin heavy chain, and then you slide actin with the labeling. So, the labeling actin will mobilize in the slide, and then you add the mutant or the wild type, the C0, C2. So, over the time you increase the concentration. At one point the mobility will be arrested. So, based on how much concentration of either the wild type or mutant needed, that can tell us the velocity of the actin fragment. And then last we have the CD spectroscopy, in which we can see the isothermal calorimetry. So, you can increase the temperature of the protein and then see the CD spectroscopic value and how they behave. So, again you can see the blue is the wild type and the red color is the mutant. There is a clear difference in comparing the wild type. The mutant protein behaves very differently, indicating that the mutant protein has lost its interaction or significantly reduced its binding affinity with the myosin S2 region. So, again so we published this one in last year. So, again this is an example for you on using in vitro experiments testing the nature of the mutation. Coming back to the organoid now, so this is Darshini Desai. She was my graduate student. She graduated last year. She is doing a postdoctoral training in New York right now. So, she hypothesized that cardiac organoid are a novel model for hypertrophic cardiomyopathy. We can study hypercontraction in vitro. So, the protocol is right here. So, you take iPSC. You can we have the agarose mold, like a spherical structure mold, in which you plate the iPSC cells, and then you can challenge them over the time with the differential media, and then in 18 to 20 days you will have a beating organoids. So, the beating organoids are almost like a spherical shape or similar to closer to spherical shape, depends on the mold you are using. So, here is an example I showed you 2 days ago. They beat so nicely. So, this is like unbelievable, like you put the cells together with all the differentiation media, you can see their beating, and you can measure the force of the beating, the time intervals between 2 beats. So, lot of things you can do with just this functional organoids. So, you can do staining, you can confirm the expression of all the sarcomeric genes, protein, you can compare that to the 2D iPSC derived cardiomyocytes. You can also do electron microscopic analysis, you can section these organoids, you can look sarcomere structures, whether you have a matured sarcomere or not within the organoids. So, this is the spatial transcriptomic analysis. So, again you can take sections of the organoid and then you can stain with various transcript. You can do really like an RNA-seq and then identify all the cell types that within that one organoids. So, again we were able to see endothelial cells, fibroblast, cardiomyocytes, macrophages and immune cells. So, which are really like almost like a human heart, where you have all these cell types. So, the question we asked was, is 3D cardiac organoid is a better model than a 2D iPSC derived model? So, we compared RNA-seq data between the 2D and 3D organoids and cardiomyocytes. Here are some of the data we showed. In 3D organoids, definitely there are some upregulation, significant upregulation of all the cellular components compared to the 2D. So, more likely matured cardiomyocytes like structures. Again, down regulation of in 3D organoid, this is the neuron developmental genes. So, lot of down regulation in the 3D organoids. Then upregulation of biological process in 3Ds. So lipid metabolism, ion channel, muscle system, proteins, and then catabolic process, oxidative reduction process, fatty acid metabolic. These were all upregulated in the 3D organoids compared to the 2D organoids. So again, downregulation of other non-significant cell, the RNAs in the 3D organoids compared to the 2D organoids. So overall, we confirmed that 3Ds are much more better model compared to the 2D iPSC derived cardiomyocytes. And then the next one, we were able to quantitate the amount of mRNA wild type versus mutant, how much they express in the organoids. So here you can see it's almost like equal level as it is a heterozygous. And then we were able to sequence, we amplify the region where you have the mutant, and this is the wild type non-carrier, you have GAC, the codon, and here we have the mutant where you have the double peak showing the aspartic acid to valine amino acid at that codon. So again, you confirm in the 3D organoid the mutation is expressed as an mRNA level. So then we used the system called ion optics. It's one of the systems normally people use to measure contractile function as well as calcium transients. There are various models that companies sell. So you can put the organoids, you can have all the peaks. Pretty much you can quantitate the calcium handling, and then you can also pace them. For example, how do you have all the organoid the same way you want to measure? You can pace them, you can give them through the transducer, you can give them power, and so you can control all the organoids at a specific megahertz or hertz, 0.05 hertz we use. And that way you can have all the same kind of force beating. From that you can analyze. Then we can measure, this is kind of standard way of measuring contractility. You have the contractile force and then you have the relaxation. So we take time to peak contract is actually 50% right here, and then we take the relaxation velocity when it goes back to the baseline. And again, the time to baseline also you can measure the distance. So you can get a lot of parameters from such a simple measurement in the system. So this is the real data. So you can see compared to the non-carrier, the mutants, that shows that time to peak 50% is reduced, which means they goes faster. It means they are hypercontraction. Time to peak 70% significantly reduced, time to peak 90% significantly reduced, and then contraction velocity overall increased in the mutant cardiac organoids. And so, and then the relaxation parameters also compare, there is not much difference here, but again there is a clear difference, time to baseline and time to baseline to 90%. We can also measure calcium kinetics using this specific dye, fluorescent dye imaging system, flow4, how faster the signal comes and then how faster it goes back. In each beat you can have this peak, and then you can calculate the transient time, how fast calcium comes in, stay in the sarcomere and then goes back to sarcom. So we didn't see any major differences between the non-carrier versus 389V, suggesting that the mutation did not affect the calcium handling within the sarcomere. However, there was a difference in the amplitude and then time to decay 50%. Again, there is a significant, but again very small differences. So the next one, we were questioning the 389V is in the C2 domain, which is very, very closer to the phosphorylation domain, where we have a number of phosphorylation sites in myosin binding protein C, and the question is whether the mutation in C2 impact the phosphorylation status. So we measure again using cardiac organoid, we can do Western blot. The top panel is the total myosin binding protein C, the middle one is a phosphorylation level. So we have antibodies against each site. Using that, so clearly you can see 275 no difference, 284 no difference, and 304 no difference when you have these are the total myosin binding protein C, but then the phosphorylation, you normalize the phosphorylation to total, and there is a difference in 275 and then 304, but not the 284. And then these two are the protein kinase C and protein kinase A phosphorylation sites. Again, it helps you to understand how the mutation can alter the phosphorylation of myosin binding protein C at baseline conditions. So this could cause the increased contractility. When myosin binding protein C is highly phosphorylated, that related to increased contraction. Then we performed RNA-seq from the organoids. Again, we were able to confirm that compared to the non-carrier, there is a clear increase in cardiac contractile oxidative phosphorylation and also hypertrophic genes, including myosin heavy chain, light chain 2, and then the NF4. So these were all upregulated in the cardiac organoid carrying 389V. So again, all these pathways were confirmed that the 389V is a model for hypertrophic cardiomyopathy with upregulation, downregulation of various signaling pathways. So because we were able to see in the RNA-seq, there is a difference in the reactive oxygen species pathway. We were able to stain the organoids. So here compared to the non-carrier, you can see there is much more ROS generation, and that was quantified here. And then also the membrane potential also significantly reduced, meaning that they are the mitochondria is highly functional to give more ATP because they have the hypercontraction. So when you have more faster contraction, you need more ATP, and that's all happening by modifying or causing the reactive oxygen species. So I think we introduced mavacamten in the last couple of days. It's the only drug that been approved by the FDA to treat hypertrophic cardiomyopathy. It's a myosin ATPase inhibitor. So in the in the hypertrophic cardiomyopathy sarcomere, you have more actomyosin interaction. So hypercontraction, impaired relaxation, altered myocardial energy. So treating with mavacamten can bring down the number of actomyosin interaction and then can reduce the hypercontraction condition. So because we were able to see that in our 389V cardiac organoids, we treat these cardiac organoids with mavacamten comparing to the non-carriers. So again, these are the baseline where you can see there is a difference between the non-carrier and then 389V, but when you treat them with mavacamten, you can bring the contractile force down significantly. Here also the time to peak is improved both 50% and 70%. So there is again going back to my introductory slide, the in vitro models or experiments can help us to narrow down the mechanism, the pathways, and also translational aspect. So again, we were able to confirm that 389V, the mutation reduces its binding with the myosin S2, and that could be the potential mechanism for increasing contractile force, and then phosphorylation, and then increased oxidative stress. Treating them with the mavacamten can be beneficial. So that's kind of thing we have done in just in vitro experiment. So what are the future directions and studies again in vitro experiments? So right now we are using antisense oligonucleotides. So what is antisense oligonucleotide? They are very small, like 10 to 14 nucleotide, synthetic. They can bind to RNA and then block the expression of the mRNA or block the translation of the mRNA to protein. And they can be very specific to the gene of our interest. So using the antisense oligos, so again you can see there are a lot of FDA approved antisense oligos already in the clinic. Patients can take some of these listed here and to treat the diseases. So we take that as an advantage and try to come up with an antisense oligos against the 389V mutation. So we were able to synthesize the region specific antisense oligos. So again it's a proof of concept study. We have a control here. We tested various antisense oligos. So here we have the wild type allele, mutant allele, same level of expression. When we treat the mutant, you can see that there is a significant reduction in the mutant, but the wild type is not altered. So we tested various antisense oligos. Again, confirm. We also have a scrambled one to show that the experimental protocol is working. Whatever we observe is purely because of specific antisense oligos, not because of the experimental artifact. So again, other panel confirming that the mutant is very, very specific. It won't impact on the wild type, whereas when you use the wild type antisense oligos, that can impact the wild type, not the mutant, and then scrambled again as a control. So overall, so what we looked into this today's this lecture is what are the in vitro experiments that we could do in the lab to test the impact of the mutation. They are protein-protein interaction studies or ATPase, functional studies, or the iPSC-derived cardiomyocytes, which is more direct, more translational. You can use this model today as per FDA. You can convince for a clinical trial down the road. Again, I talked about challenges in organoids. You can also expect some variabilities, and again batch to batch variabilities. Co-culture, for example, you have to have not just cardiomyocytes, but also consider to include fibroblasts or endothelial cells, and then how that adding the other cell types into the organoid can modify the function. So these are the challenges in using cardiac organoids. So again, I want to thank and acknowledge Darshini Desai. She was my graduate student. She performed all the cardiac organoid. When we started the project 5 years ago, when I told her, this is your PhD work, your job is to generate cardiac organoids and characterize them. And it was a challenging time because 5 years ago there was no protocol, not much been known about developing cardiac organoids. Within first year, I would say she was like, one day came to my office, oh my God, Sakthi, it's not easy, I'm struggling, I don't know whether I can make. But then she pushed hard and finally she made through the protocol and established a nice model in the lab. That's all I have on the in vitro models, and I'm happy to take any questions you guys have. Thank you. Questions on in vitro models. How do you select an experiment? What kind of in vitro model you can choose? A lot of things you should decide before you start any experiment. Again, based on what you have in the lab, what you have in the department, or next to neighboring departments, or closer to you, or sometimes you can collaborate. You can ask anybody in the world, can you do this one experiment for me? You know, if somebody has an expertise in FRED technique or you know, mass spec, if they have do mass spec routinely, you can send your samples. So everything is possible, but again you have to start with somewhere convincing other people to do the experiment for you. Any questions? Yes. Can you use the mic?
Some kind of excitation to the
You're talking about the 2D culture or 3D culture, right? So you culture them, you change media every day or alternatively. At one point you have to add differentiation medium. That differentiation medium is so important to differentiate what cell type you want to have, right? So once you have them, and then within 2 weeks, in our case, the cells will start beating, okay? And so from that point, you have to keep maintaining the medium with giving all the resources the cells needed. You can culture them up to 80 days, 100 days. But again, you don't, depends on the nature of the project, the question you are asking. In our case, we stop them at 25 days and then complete all the studies. Again, you have a control and you are comparing your experimental one with a control one all the time. So you are looking all the differences. Yeah. So again, based on the question you are asking, you can decide your experiment.
We also see some kind of mechanical binding between the cells.
Yeah.
And what are the mechanical nature that you look for?
Yeah. So when you plate the cells, you have to know the numbers, how many number of cells you are plating, right? If you put too many cells, then it will come quickly confluency, right? The more cells and they attach, everything attach, and then you can't really do experiments. So you have to start with minimal number of cells. When they mature in 2 to 3 weeks, then they occupy the empty space. So you have enough space for individual cells to enlarge and beat. If you overload now more cells, then you don't know what cells is giving, you know, lot of compound effect will be there. So you want to measure single cell impact rather than compound cell impact, you know. So we try to isolate once the cells are matured by, you know, cell separation. It was very hard. These cardiomyocytes very sticky. It was very, very hard. Whereas in adult, in mouse heart, we do the perfusion system. We perfuse with the collagenase, and then we dissect everything, and then we can isolate rod-shaped cardiomyocytes. But here it was very, very difficult.
Sorry, I am getting more.
Yeah, no, yes.
So the thing is, if the separation is really a problem, I think I am loud enough to.
I think they want to go in the record, so you know, yeah.
If there is a problem with the separation, so can you use the micro channels, some kind of like very small channel where you can pass just one cell or one tissue and then culture it so that you don't have any problem with the separation, and then you can also see there is not binding with the other cell as well.
Yeah.
So I think
Yeah, it's
that
great,
can be
great question. It was done already. People have tried it and showed that the maturity, everything. There are a lot of advantages. So again, based on the resources, the time, a lot of factors influences your experimental design. If somebody published a paper on such a, and then you try to replicate, you spend time on it, right? Sometimes it won't work, sometimes it works. So people, what they do is they start their own way of doing experiment and then get the work done. Yeah, but people have done it. Based on the mold or the shape they give, the cells will change it. T shape, L shape, different types. Yeah. That's a project for biomedical engineering students. Yes.
So these iPSC cells are heterogeneous or homogeneous in nature, sir? Genetic.
So they are heterozygous. Again, based on if you collect the sample from a heterozygous subject, they are heterozygous. So you have wild type allele, you have the mutant allele. But if you collect the blood sample from a homozygous subject, then you have both are homozygous mutation.
Okay. All right. And there was one more question, sir. The RNA-seq data that was shown in your slide, it was for the entire organoid tissue.
One organoid, yeah.
Okay. Did you ever had an opportunity to go for single cell RNA-seq? Because
that's what I was telling. My students spend a lot of time try to dissect the organoid and collect single cells from the organoid. It was very, very difficult. So that's what we performed the spatial gene expression, where you cross section the organoid and then you look for all the RNA expression. So we were unable to isolate single cell from the organoids. Very good question.
Hello, sir. Sir, you studied the deletion and tried its effect. Wanted to see the effect on various aspects of the organoids. In the process, you looked at the transcriptome and tried to know what are the pathways those are upregulated or downregulated. On the other hand, you had also shown that organoids had an elevated level of ROS, and that you showed by microscopy. Was that data correlated with your transcriptome analysis? Was there any kind of overexpression of oxidase or peroxidase kind of enzymes which actually are implicated in a elevated ROS generation? And what you showed was the image, microscopic image. What was the method of calculating the overall elevation of the level of elevation of the ROS in the organoids?
Yeah, so again we performed the biochemical staining approach to look for the level of ROS production. We also performed, I didn't include the data, it's in unpublished data, we measure mitochondrial function using seahorse experiment. I don't know whether you know about the seahorse. You can look oxidative whole the various stages of oxidative process of the mitochondria. We found that the 389V are much more, they need more oxygen consumption, producing more ATP. So clearly there is a demand and supply. You have hyperfunction, meaning that you need more energy, right? So the ATPs are the one has to supply the ATP. So there is an oxidative stress is increased. So again, constant ROS production is also not beneficial. It's detrimental down the road. They could cause inflammation, etc., and part of the disease development.
I have a technical question, sir.
Yeah.
In your experiment, may I know how did you measure the ROS that was being generated? Just technical.
I think in the biochemical assay, we stain the cells with a specific dye that will observe the ROS produced. So the intensity is the outcome. So I don't know top of my head.
Okay, so the,
I can send you the paper. It's in the paper. Yeah.
Thank you, sir. Thank you.
DCH. Yeah, I think so.
Hello. So it's very fascinating to see the heart, like organoids are beating. So, like how well developed the actually I have very little knowledge about the organoids culture. So, what level of development is there in the organoid? Like, do they also develop the vasculature or neurons or something?
Yes.
It's well developed.
So again, organoid, you can develop the way you wanted. You can give proportion, number of cells like cardiomyocytes, how many cardiomyocytes, how many endothelial cells, how many fibroblasts.
Okay.
You can change the composition of the total number of cells to given to the organoid. Based on that, you can create all these types.
So you have to like give all the components.
Yes.
Whatever you want to develop, and then they will assemble into one organoid.
Yeah, and then see whether, so again in the heart 75% are cardiomyocytes, 25% other cell types, right? So without having the 25% cell type, if you look only the organoid with the cardiomyocytes, you can see exactly the impact of the phenotype. But sometimes if you add the other cell type, that changes the outcome of the phenotype. But you know, that could be more closer to human heart, you know. So.
And also, since now they are consuming more energy because the mechanical things are going on. So, do we need to supply ATP or they generate enough ATP by
So, they utilize more, they burn more fatty acid. Yeah.
From the media?
Yeah.
Okay.
Yeah.
And one more, it's a kind of detailed question about this S2 and C0C2 interaction, that part only.
Yeah.
So, when you express this S2 in E. coli or in bacteria only, so do they assemble into coiled thing or it's only one helix?
I think it's assembled coil, but again we don't know. We never did any cryo-EM
or any structural.
Yeah, not structural.
Because they tend to form, they can also form filaments.
I think they form coil because that's what the CD spectroscopic data showed, the isothermal calorimetry assay.
No, that is for binding, yeah. I mean, ITC is mostly for binding, so maybe if it is binding, then it is forming the coiled coil, maybe.
Maybe not, I don't know. Maybe not enough for forming the coil.
Oh, okay.
I don't know.
Okay, thank you.
Hello, sir. Earlier you said that you have done the cross-sectioning of organoids. So, what is the average size of organoid you have made in your lab?
What?
So, average size of organoid. Size.
Oh, average size. Again, I don't know the number exactly. Few micro. It's very tiny. You cannot look at a naked eye. You need a microscope to look at it. To process this, again, we outsourced. A lot of companies now in California, they do the spatial mapping. So, in our study, we gave them the organoid, fixed organoids. It costed us almost $15,000 just for sectioning and doing the transcriptomic analysis. So, because we want to be a pioneer in doing this, so we don't have all the facilities in the lab, but so we outsourced a lot of things. Yeah.
This was in continuation with the explanation
of
yours, sir. Didn't you feel able to do MARDI imaging? I mean, a technique wherein instead of the sections, the thin sections were subject to the MALDI top. So a special imaging of the metabolite in this case.
Yeah.
There, special.
Yes, you can isolate total proteins. You know, you can do the top down or the mass spec, right? Proteomics. You can do. You can look. Yeah.
My point was that since the sections were sliced,
yes,
and RNA-6 of these sliced sections were being performed,
yes,
where the aim through your slide was to know the express genes, and you were mapping the spatial distribution of the expressed gene in the organoid.
Yes,
Yeah.
what are the cell types in this organoids having?
Yeah. So there was a technique called as MALDI imaging.
MALDI imaging, okay.
Yeah. So
No, I'm not familiar with that.
okay, so
So it's kind of, you are going through proteomics approach.
yeah.
Okay. So you can do the same way, you
Same
section
way, spatial distribution of the
special
protein.
distribution
Yeah.
for proteins.
Yeah.
Okay, very good. Good to know that. Any other question? I think we can have 5 minutes bio break and then we can reassemble again. Thank you. Hello sir, how are you?
Fine, how are you?
Almost ready for the weekend.
till at 1 o'clock. So what
12:30 will be done.
Yeah, so another 45 minutes and then tutorial.
No, another 45 minutes and a tea break.
Tea break.
And then tutorial. 12:15 will be done. I will make it sure. Okay, let's get started. Before we go to the next session, do you have any questions for me? During this break, I'm sure you discussed something and come up with some questions. Any questions I can answer before we go? Yes, please. Questions are always welcome. So we have to start. We have to start with in vitro to say, like, a lot of the experiment, right? Simple experiments, you can get it done. In vivo takes time. So as we know, right, you create an animal model, takes years and years. So while you are waiting, you can complete all the in vitro experiment to justify why we need animal model. For example, in the in vitro, if we don't see any differences, then how are you going to justify you need an in vivo model? You are putting time, you are putting money, right? And then you are going to ask a funding agency to give money. You need to justify. Or if you don't have any result, like showing differences between the mutant and the wild type, you need to justify why we have to create animal model. Unless you have plenty of money, then you can just generate a model and then you can study. But normally, the in vitro experiment is a starting point. You can easily get it done with the available resources in the lab and then justify to go to the animal model. Even for our 25 base pair deletion, we first published a paper in JBC just to show all the in vitro experiment. And then we generated a mouse model and then we asked for money. In the mouse model, you show preliminary data with the phenotype and then you ask for money. I want to characterize this model. I want to show the phenotype. I want to treat this disease, etc. Then you get the money and then you do 3 to 5 years time to study the mouse model. And then we published that one in the cardiovascular research, which is a 12 impact paper. So you go step by step, again based on how much money you have in the lab. Most of the labs are middle class labs, and so we start with the in vitro experiment. Again, once you know that it's big difference in the phenotype, this is an easy start. But at the end, even though you have an in vivo model, you have to come down to do the in vitro to justify, to give the rationale and what mechanism is involved. Is it myosin heavy chain is activated, or how do you tell? Only by measuring ATPase activity, right? Yeah. Okay, any question? So going to the in vivo model, again, just want to see the differences. So you have the normal heart and then the hypertrophic cardiomyopathy where you have more thickened wall, and then the dilated cardiomyopathy or heart failure, your walls, the ventricular walls are very thin and then the chamber size is so big. Pretty much end stage heart failure, you need a transplant. So why are animal models important in hypertrophic cardiomyopathy research? What do you think? Why animal models are? You said, why don't we go directly to why? Why in vivo models are important? Why can't we just conclude with our in vitro model, this is the mutant and this can cause phenotype, done. We can start the treatment.
Because organism as a whole is composed
It's,
different systems that comes together to
yes.
form out the.
So it's a physiology, right? You want to see the whole animal level, organ level during the development, how the disease can be prognosed. So, importance of animal models, number one, we are replicating the human condition in an animal model. Exactly, we are introducing the mutation either through knock-in or knockout or transgenic overexpression of mutant protein and then mimicking that. So, we can study a lot of these properties only in vivo, not in the cell culture experiment. Hypertrophic mechanism, fibrosis, arrhythmogenesis, whether that can develop left ventricular outflow tract obstruction, which is the case in myosin heavy chain mutation, and then heart failure. So, again hypertrophic cardiomyopathy, as you know, have you seen the last 3 days, it won't happen overnight, right? It's not like infectious disease. Get the infection, you 1 or 2 days you see the impact, but this takes years and years and years. In some of the genetic mutations, it takes more than 50 years, 60 years. They cause late onset. Some diseases, some variant can cause early phenotype. So, definitely the time point in the disease development only can be studied in vivo, not in vitro experiments. So again, when you look into all the pathways in the myocardium or in the cardiomyocytes, there are a lot. We already have number of drugs, small molecules that being used right now. For example, we talked about the beta blockers, the calcium channel blocker, ACE inhibitors. So there are a lot of already been in the market, in the clinic to treat heart failure. Now we have mavacamten to treat hypertrophic cardiomyopathy. So we have to have a preclinical model to test all these various drugs and see whether that can be translated into the bedside. So you start with preclinical model, again you validate everything in vitro, and then you go to the animal model where you can study safety and efficacy study, and then you go to the clinical trial, and then you go to the final FDA approval. Again, it's a very simple diagram, but each step there are a lot of steps to be completed. So animal models of cardiovascular disease. So here I listed starting from mouse model, but even zebrafish, Drosophila, they also been used in studying hypertrophic cardiomyopathy or heart failure, heart disease as an animal model. But again, at least a mouse model, it's very close, better models than the zebrafish or the Drosophila models. Why are mouse models are predominant? Again, you can take all the models, only being mouse models being the predominant model being used in the literature. So number one, genetics are relatively well understood. We know the genes, all the genes of the mouse, well characterized, well known. There are various strains of the mouse. This is the FVBN strain, white. There are BALP-C, C57 or 126. There are so many different strains. Again, remember that these strains can also give different phenotype outcome. The same mutation, if you put in different background, you will have a different phenotype outcome. So you have to again know what you are selecting. So and then the genes are almost the same comparing to human. We can generate the colonies so faster, defined cardiac mechanics and hemodynamics, and then this offers more preclinical tool to study any treatment options or toxic effect, etc. So again, as I said, the development, disease development can take time. So if you have an animal model, you can really look the heart every month by echocardiogram, right? So you can really look the developmental process by doing echocardiogram. This is a non-invasive. We have a small echo machine that can be used to measure mouse cardiac function, and so it's a, we can define at what time point the disease is now starting, you know. So there, in the literature, if you look at the literature, a lot of mouse models already available. So transgenic are very common in the early time before the CRISPR was introduced, and then we have CRISPR-Cas9 genome editing is now. The transgenic is like cardiac specific transgenic is we use alpha myosin heavy chain promoter and then tie the gene of interest with the mutation, and then you have the human growth hormone polyadenylation sequence to have the stable mRNA in the sarcomere or the myocardium. So all these approaches were used in the past to generate mutant mouse models for myosin binding protein C and myosin heavy chain. The knockout models are being used to study haploinsufficiency. Again, you know, if you have one allele wild type, you don't have the second allele, you can, thus one allele is enough to produce 100% maximal protein when you are young, but aging process can cause haploinsufficiency or presence of any secondary risk factor. Again, in vivo, the beneficial of in vivo is you can test all the secondary risk factors, challenging the mice to diabetic, challenging the mice to hypertension, you know. So you can study all those secondary risk factors using the mouse models. The experimental technique, so all you have to have at least echocardiography so that you can measure the wall thickness, cardiac function, and if you have MRI, some institutions in the States they have MRI, they can also look at the mouse heart to see the fibrosis and contractile function as well. Then we have the pressure volume loop that also can measure the contractile, all the fine parameters of contractility. We can do histology, fibrosis, myocardial disarray, and then we can isolate RNA, protein, look single cell RNA you can do, single cell proteomics you can do. So a lot of things you can do from these mouse hearts. There is a very nice article this year, and I will send you this paper. This is in 2025 April. They covered experimental models of hypertrophic cardiomyopathy, a systematic review, a very nice review. You can see it's almost 30 pages. So again, it's a very tiny figure, but maybe here you can see. So the studies using mouse models, they compare from 1995 to 1999 to all the way. I think 2010 to 2014 has more number of papers using mouse models. Gene of interest, myosin heavy chain, already 70 articles been published with the mouse model, and then second one 60 with myosin binding protein C, titin 2, troponin C, troponin I, and then troponin alpha tropomyosin, then other sarcomeric proteins. Here countries of publications, you can see the, I mean, most of them from the States, and then also from China, some from Australia, and then the European countries. I don't see anything from India here. There are some studies. Okay. So I'm sure, but maybe very small, couple of papers probably. The mouse model sarcomere gene is the predominant 80%, and the rest of them are non-sarcomere or compound heterozygous etc. combination. Again, the mouse models being used, hypertrophy, for example, if there is a model, what is the percentage in hypertrophic or the phenotype? Yes, here for example, myosin heavy chain, you know, a large number, myosin binding protein C large number, but there is a percentage have no phenotype. So again, when you generate a mouse model, it doesn't mean you will see a phenotype. It depends on the mutation, depends on where the mutation located in the gene, whether it's impact contractile function or not. Sometimes you don't see any differences between the wild type. So what you have to do is you have to challenge the mice to some cardiovascular stress, and then you can see the differences. So again, you have to play around if you don't see a phenotype at baseline. Maybe you can age them, you can see up to 1 year during the aging process, maybe you will see a phenotype. Sometimes some mouse models, they can go up to 1.5 years, 2 years, meaning that you have to spend a lot of money to pay for the colony, you know, the cage. So in our university, per cage is, per day is 75 cents. We can have up to 4 mice in one cage. So I have almost 350 cages. So per day you can imagine how much money we pay. So sometimes you see the mice, they fight, so you have to separate them, you have to hold them one single cage, you have to pay for the same cost. Whether you have 1 mouse or 3 mice, 4 mice, the cost is the same. They calculate based on number of cages, not number of mice. So again, these are the very nice articles I can send you. You can read more about the importance of the in vivo models and what are the models are in the literature and etc. So I will give you some examples from our lab. So this particular promoter, alpha myosin heavy chain promoter, we used to overexpress gene of interest in mice. So the question is, why do we use alpha myosin heavy chain promoter for a transgenic model instead of beta myosin heavy chain gene? So we have 2 myosin heavy chain, right? Alpha and beta. We use alpha in mice to overexpress the mutant protein or gene of interest. The question is why we don't use beta myosin heavy chain. Okay. Developmental stage. Okay. So, so again we have 2 different paralogs, alpha and beta. In human, beta myosin heavy chain is the adult form. Alpha is an embryonic form. You know, after birth we have beta myosin heavy chain. Before birth we have the alpha myosin heavy chain. In mouse it's just opposite. We have a beta myosin heavy chain before birth in mice. After birth we have the alpha myosin heavy chain. So the switch occurs. Alpha myosin heavy chain is called faster myosin heavy chain because it hydrolyzes ATP very fast. Beta myosin heavy chain is a slow myosin heavy chain. It hydrolyzes ATP slowly compared to the alpha. Why this difference? So in mouse, as you know, in a minute it beats 500 to 600 times, so fast. So you have to have alpha to support that number of beats. In human it's 72 to 75, so we are switching, we are slower and we have the beta myosin heavy chain, and so that's why. So if you have a mutation in beta myosin heavy chain, you develop hypertrophic cardiomyopathy, but if you have a alpha mutation in alpha myosin heavy chain, mostly congenital and then also early dilated cardiomyopathy or heart failure, you know, because alpha don't expresses after birth, right? So that's the reason we have to use alpha myosin heavy chain promoter And then we have a MIC tag. This is a 10 amino acid human tag. We put it in front of the cDNA of our interest so that we can differentiate the endogenous protein versus exogenous protein. So, when you collect total lysate, how do you differentiate what is your transgenic protein versus your endogenous native protein, right? So, this tag can be used. And then we have our gene of interest, and then we have the human growth hormone polyadenylation sequence. That sequence is critical to have a stable mRNA in the cells. Otherwise, the mRNA will get degraded very fast. So, using this promoter, you inject into the embryo and then you generate the mouse. It's all the core facility. We do not do the entire process in the lab. We just outsource the core facility in the university. They generate the model and they give us the model, and then we start characterizing them. So, again, this is an example. We published a paper in 2019. Expression of the mutant protein in the myocardium definitely cause hypertrophic cardiomyopathy phenotype, and so now we are using this model to test the gene correction or antisense oligos, whether we can reverse or improve the cardiac function. This is another example. People have now generated the pigs. These are the small, not the larger pig model. And so again, the myosin heavy chain gene was mutated. Here you have the beta myosin heavy chain, not in alpha myosin heavy chain. So again, mouse, rat, adult has the alpha, whereas rabbit and other upper animals, they also very close to human. They all have beta myosin heavy chain in the adult. So that's very good. But again, breeding rabbit or pigs or higher animals, more expensive, more time consuming process, and etc. And you don't have to have enough N number, you have to do a lot of breeding. But there are models. So this one, we published this paper with the Maine Coon cat. So it was very interesting story. So in the States, families, they own Maine Coon cat as a pet animal. You know, these Maine Coon cats are very large. They are really like a dog. You know, very big, and they have hypertrophic cardiomyopathy. Very natural selection. It's not artificially you introduce the mutation or not. And one of the families, they looked into the literature, who is here in this local studying hypertrophic cardiomyopathy. They found my name. And so they contacted me saying that their Maine Coon cat, through their veterinarian, saying it has hypertrophic cardiomyopathy and they want the cat to be treated. So they contacted me. I said, okay, I'm not a cardiologist, number one. Number two, I have to know the genetic cause for this disease. So I need to test whatever known mutations are. I can do it by simple PCR assay. I can decide, I can look, and then I can tell, okay, this is the mutation, this is a gene. That's all I can do, you know. They said, okay, we give you the blood sample, right? So we had to go through all the animal protocol, approval committee, and then material transfer agreement. You have to have everything. I mean, you know, these are very sensitive, you know, issues. So if anything you do wrong, it goes to media and then you will be in trouble. So we got all the approval, and then we collected the blood sample, and then we tested for all the known gene mutations. There are not many for a Maine Coon cat. And then we found none. So we told them, we don't, this is probably the novel gene, novel mutation. All the known is not present in this Maine Coon cat. And they said, okay, how much would cost for you to do the whole genome sequence? Yeah. So I said, probably including bioinformatics, everything, 3,000, 2,000 to 3,000. Okay, we give you the money, please do it for us. Okay, then we performed the whole genome sequence of the Maine Coon cat, and then we identified the mutation in the troponin T. Then we said, okay, to conclude, we need the family. it's not, maybe it's sporadic, right? So then they went back to the breeder. The breeder is in a different city, and the breeder was unhappy because, you know, if they come to know they are using this model, then they cannot sell. It's bad news for them, but they were very coordinate, and they give they identified all the siblings and parents, and you can see that how they are sister, and you know, they come from here. The mother, the son is kind of the father of the these two cats, you know. So it's a very unique way they breed. Anyway, so this is a homozygous animal. So again, it has a clear phenotype. So it was a very nice story, but again, this is the natural model. Now, after seeing this publication, AstraZeneca contacted us saying, okay, we wanted to test our molecule in this model, you know. So we are trying to collaborate now with AstraZeneca. So again, this is the way you kind of build your research. So we talked about hypertrophic cardiomyopathy versus heart failure. So in hypertrophic cardiomyopathy, you have increased force, increased contractile function, whereas in the dilated cardiomyopathy or the heart failure, you have decreased cardiac function or less force overall you generate. So here you have to use inhibition because you want to reduce the force generation, and here you have to use activator so that you can increase the force. So it's kind of two different way to treat hypertrophic cardiomyopathy. Again, we are going back to the sarcomere structure. We have titin, we have thick filament proteins, myosin heavy chain, myosin binding protein C, and then thin filaments, actin, troponin complex, and alpha trophomyosin. So myosin binding protein C we call as a brake, as you can see in this picture. It's tightly interact with the myosin heavy chain and control how much this interaction can be occurred so that the force can be generated. So this interaction is controlled by myosin binding protein C interactions. It can happen only when it's get phosphorylated. So myosin binding protein C gets phosphorylated only when you are hyperactive. Again, I told you, when you have increase in heart rate, this protein is highly phosphorylated, so that it goes away from myosin heavy chain. So the myosin heavy chain can interact with more actin and generate more force. So now so a lot of companies are now so much interested in studying these interactions and then modulate the interaction so that you can treat hypertrophic cardiomyopathy. Here again I want to show this is the myosin heavy chain protein, very large protein, 240 kDa. It has the essential light chain, regulatory light chain, different regions, and then the cardiac myosin binding protein C, all the interacting domains. So this is a study we performed in our lab. So we published a paper in JVC 2021 specifically in the S2 region. So most of the literature covered a lot of mutations in the head regions, but less interest was given to S2 region. This is the important region actually. When you enlarge that, you can see how many mutations being reported just from this region. You know, these are the mutations are known causes hypertrophic cardiomyopathy. So we used, so again we have to use the animal model to study various secondary risk factors. So in this study what we try to come up with is identify the interacting peptide, the region, number one. Number two, how can we break or we introduce the break you know, between the myosin S2 and then myosin binding protein C. One is phosphorylation, right? When you, this is a dephosphorylation, when you phosphorylate C protein, that interaction is gone. So now we propose that can we introduce a peptide in between this interaction and can break the interaction. So, we want to identify a small region of myosin S2, that exactly region that interact, and then use as a peptide therapy to break the interaction. So, in heart failure we have increased dephosphorylation. The more dephosphorylation you have, reduction in contractile force. So, there are a lot of peptide therapies already in the market. So, here I have some examples. Several diseases they use proteins or peptides. So, why not peptide therapies, right? So, this is where Rohit, he tried to identify a small region of myosin S2, and that can be a potential peptide to treat either hypertrophic cardiomyopathy or the heart failure. So, he used two different assays. One is a binding assay, another one is in vitro muscle assay. I will go through that one by one. So, here when you express the recombinant protein and using HISTAC, you can purify the protein of interest in so clean. So again in bacteria you have so much protein, but you overexpress the mutant protein or protein of interest, and then using HISTAC column you can purify. You can see how enriched you can have. And then here you have the C0C2 recombinant protein. So, this is the binding assay. You can coat the S2 and then you wash, and then you add the C0C2, then you wash, and then you put the antibody, and then you wash, and then you put the secondary antibody, and then you develop the color. So, he developed a lot of peptides in overlapping peptides of myosin S2 region in collaboration with Amgen and the company. He found that these three peptides, 23005, 23006 and then 27119 show very nice concentration dependent decrease in the signal. So, the more binding you have less signal, because the peptides compete with the C0C2, meaning that the C0C2 will be removed by washing. So, and then you have less signal. So, that's the way you calculate. Then we use, this is another major in vitro experiment we do. So, called P-calcium, calcium force measurement, right. So, we isolate papillary muscle from the heart, and then you can skin them. What does that mean is you can treat the papillary muscle with kind of detergents, so that you can remove the membrane, all the cellular components. You just have only the sarcomere proteins. So, here you can see after the skinning, they are very transparent. You can see the striations actually very nicely. And then you can attach them into these clips, and then it goes into the motor, and then you can measure when these were in the small wells with a buffer. So, you can increase the calcium concentration, and then you can see the force generation. So, this is the sigmoidal curve. This is the maximum concentration calcium you need to generate this maximal force. So, this is a very standard assay. Using that assay, you can look for how much force this can be generated. So, here we have a PNAS paper where we showed that this is a wild type fiber, and then the wild type fibers were treated with entire S2 protein, and that can increase the force, maximal force. And then also we were able to determine how faster the rate of force redevelopment. When you stretch the muscle, how quickly it goes back and then come back again. You can see there is a significant increase. So, it is kind of justification before we go for the peptide therapy. So, Rohit used that approach, and he again selected these three peptides and measured the maximal force. Again he identified that the 23005 and 6 as well as 27119 shows very clear force increase. When we treat the fibers with these peptides, there is an increase in force. So how do we translate this peptide in vivo? So that's a question. So we have a model, mouse model, where we have mutated the phosphorylation sites to either through CRISPR-Cas9, either alanine mutation or aspartic acid mutation. So this is the phosphoablation, this is the phosphomimetic. So you can see here the phosphoablation show heart failure. They have reduction in cardiac function, enlarged heart chamber, etc., whereas the phosphomimetic is kind of normal in cardiac function. So Rohit isolated papillary muscle from these animals. Here we have the wild type, then the heterozygous and homozygous of the phosphoablation, and then see that there is a reduction in maximal force of this fiber. Again, the phosphoablation means you have dephosphorylation of C protein. That means it has more break on the myosin heavy chain and less force being generated. So using that model, we treat in vitro again and then challenge the fibers with my S2 protein and then see whether that can rescue the phenotype. Again, we were able to see that compared to the control, treating the fiber with the S2 protein, there is a significant improvement in the force development. So you can break the interaction of myosin S2 and myosin binding protein C using the recombinant proteins. Again, these are the beneficial of using mouse model or in vivo model. You can validate any drug development if you want to do. So going back to the mutation story, so we have a paper in JBC how the S2 mutations that can impact the interaction with the myosin, cardiac myosin binding protein C and then cause hypertrophic cardiomyopathy. So again, I showed you there are a lot of mutations in this region. We selected two mutations, one is a missense mutation, another one is a deletion mutation, and then see how that impact contractile function and developing hypertrophic cardiomyopathy. So we have now a mouse model using CRISPR-Cas9 approach, a knock-in model, introduced the glutamic acid 903 deletion, which is in human beta myosin heavy chain, whereas in mouse we have it in alpha myosin heavy chain. The amino acid sequence is the glutamic acid 932, and we removed that as a deletion. So that's exactly this region, and in families with this mutation is pathogenic. They show very severe disease, and so we selected this particular mutation. And here is our mouse model. These are within 6 months to 9 months of age. The deletion in myosin S2 region causes hypertrophic cardiomyopathy phenotype. They develop increased in heart weight, body weight ratio, and they also started developing heart failure after 6 months of age. Clearly, there is a reduction in cardiac function. We also have cardiac organoids, so for the same mutation, but in the beta myosin heavy chain, because this is from the human iPSC line, but isogenic line. Again, it shows there is an increase in contractile function, kind of replicating the phenotype of hypertrophic cardiomyopathy. From the mouse heart, we can do RNA-seq. This is Kalyani, my postdoc. She performed the RNA-seq. Again, you can see a lot of hypertrophic genes are upregulated in the heterozygous mouse heart compared to the wild type or non-transgenic mouse heart. This is a graduate student. She started this project. She validated this RNA-seq by doing RT-PCR individually on these genes, confirming that they are true. The RNA-seq is correct, validated, and there is an increase in hypertrophic markers. We also isolated the myocytes from this mouse heart. The myocytes can be skinned, removed the membrane, everything, and again you can challenge them to various calcium concentration. You can measure the force, how much the myocytes can generate. Again, you can see compared to the wild type, the mutant myofibrils show more force compared to wild type. So, here is a summary of this maximal force, and that is very significantly different. We also can isolate the fresh cardiomyocytes. These are not skinned cardiomyocytes. You can, once you isolate these cardiomyocytes are live within a day, you can measure contractile force and calcium transients. Again, comparing to the wild type, there is an increase in cell shortening, and then also there is a relaxation velocity also decreased here. So, overall these mouse models are being used to verify and validate the development of hypertrophic cardiomyopathy, and also use the iPSC's in vitro models to crosstalk. Again, I indicated that if you want to test the presence of secondary risk factors, there are a lot of ways to do that. So, for example, if you want to introduce pressure overload, we can perform surgical model, aortic constriction, and that can generate partial occlusion of the artery, and then the ascending aorta. So, and then you can develop more pressure in the heart myocardium, and that can cause hypertrophy. We can induce a myocardial infarction, ischemia, reperfusion injury. This is the model which is very close to human, the reperfusion model. And then toxic, we can inject doxorubicin or isoprotrinol, angiotensin, etc., and then see how that toxic condition cause the heart failure. Or crossing the mouse model with another mouse model carrying a mutation. So, you have a double mutation, how that can impact. Here is an example, like this was my PhD work. I was doing surgery on rat inducing pressure overload induced heart failure. It's a very standard technique. You partially occlude the aorta, resulting that you put more pressure into the myocardium, and that could develop hypertrophy within 8 to 12 weeks of time. So, what are the challenges in the mouse model or animal model? Again, like I told you, every model there are challenges. There is no perfect model, but you have to take the best suitable model for you to test the hypothesis, to develop therapies, etc. So, again, even though we use a lot of mouse model, it is not the perfect 100% mimicking human model. There are a lot of differences and variabilities in penetrance. Again, mouse cardiac function, mouse the contractility is entirely different from human, and then now there are ethical concerns in larger animals. So, we have to really work on mouse or rats, etc. The primate model would be very good, but a lot of concerns on ethnics, etc. So, in conclusion, in vivo models are critical, important for hypertrophic cardiomyopathy research, specifically studying the genetic mutations. And again, we should consider various types of models, and comparing to in vitro, we need to integrate all the technique so that we can compare and validate all our findings are accurate. So, the future ongoings are like how can we introduce gene editing or mutation within embryo, so you can correct the mutation before the baby is born, or so we can correct the mutation very early on. So, final summary is animal models are key for various purposes. But again, it is not the perfect model, 100% not perfect model, but it is important to choose the right animal model based on the question you are asking. So, your next mouse model is only a mouse click away. So, you do not need to do it, you just pay for it. So, a lot of companies, a lot of core facilities, they just do it for you. It's a lot of time. I mean, it's a time consuming, and these days it's cheaper actually compared to 10, 15 years ago. With that, I want to acknowledge some of my lab members, specifically James, Rohit Singh, and Brendan from Amgen. He was one of our collaborators. They provided also funding support to characterize the myopeptides. So with that, I want to conclude and this session in vivo, and happy to take any question you have, and then we will have a short break for tea. Thank you. So animal model, in vivo model, why, how, what is your thought, what is your question? Yes.
With regard to the peptide therapy.
Yeah.
Yeah. So, I mean, just technical question. What or how did you decide the amino acid residue sequences in the peptide therapy to use those peptides to apply it as a brake on myosin?
Yeah, so you take a region of the binding, right? You take protein A, protein B, and these two protein binds here. So you take this region, right? And then you generate overlapping peptides, 10, 15 amino acid, 10, 15 amino acid. So you are making overlapping 50 peptides. Okay, and then you narrow down which are the peptides are really binding.
Okay, so initially a combination of the peptides were tried,
yes.
and then based on the data.
Yes.
Okay.
So once you narrow down, then for example, for the in vivo purpose, you have to attach homing peptide, another peptide that is very specific to cardiomyocyte. So once you put the peptide into the bloodstream, this goes into circulation. How do you deliver this very specifically to the cells, to the heart cells? Otherwise, you will be cleared in your liver and it's gone, you know, or it will go to skeletal muscle randomly somewhere, right? So you add homing peptide so that you can deliver these peptides to specific cell type.
Okay, so
But we are not at that stage yet.
okay, yeah,
Have you taken any help from in silico simulation for this selection of the peptides?
Yes, we have done it. I will show you the data maybe tomorrow. I have slides.
Okay.
We narrowed down to 3 amino acid through that model.
Okay.
And then we use that model to identify small molecules. That molecules also can bind similar to the peptide. So instead of the peptide now, you can put the molecule and that can do the same job as the peptide.
Okay, and that is how you ask Amgen to provide those peptides, correct?
Amgen helped in the peptide project, but again, you have a 1 year contract, 2 year contract, and now it's over. So now we have they opt out from the intellectual property, but we have the person from the Amgen, Aaron, he is part of our project. So we have the provisional patent. Now we have identified 12 small molecules that can mimic this peptide. So now we have to test the small molecules and then we narrow down and then we can go to the next patent.
And these peptides were designed to reduce the kinesis activity, correct? Phosphorylation.
Yeah, so the peptide is to break the interaction between myosin heavy chain and myosin binding protein C, particularly when you have a decrease in force, because the C protein puts more brake on myosin heavy chain. So you want to put the myosin S2 peptide that competes with the endogenous myosin heavy chain and then release the endogenous interaction. Then you have the endogenous myosin have more actomyosin interaction, generate more force.
Thank you, sir.
Questions from students? Any questions from the students, master students? Yes.
Toxic cardiomyopathy, and in front you had written dox.
Doxorubicin.
Doxorubicin. So
Yeah,
doxorubicin
it's
is
a cancer drug.
Yeah, it's a cancer drug. So, so administration of this drug is, can exert this kind of,
Correct. Doxorubicin can cause dilated cardiomyopathy. Most of the cancer people, they die not because of cancer, it's because of the heart failure, because of the drug.
So that is what is the side effect of the drug.
Side effect, yeah. So it's a lot of work is going on having cardioprotective cancer drug to overcome doxorubicin, but it's still the best drug in the market. I think Princess has a question. For students, if you ask one question, I give you 100 rupees per question.
I think maybe I can wait. For
No,
the
you can
students
ask,
to
I'll just give some time.
Okay, so about this,
100 rupees per question from students, okay?
about this alpha to beta switch, like beta to alpha switch or whatever in both the organism. So, like I have two questions. So one is the based on the rate, like you said in rat we have a reverse thing, like beta first and then alpha in adults, and in the human we have vice versa. So what about the highest like the elephant and other things? Do they have any other form of some like gamma,
I
or
don't
it is
know.
not discovered yet?
I don't know, yeah.
Okay.
Probably, I don't know.
Okay.
I don't know what elephants, adult elephant.
Like that is also 5 times lesser the heartbeat.
Yeah.
17 something.
Yeah,
So.
so we will see.
And other one.
We have here, because the Department of Zoology,
genetics, yes.
I'm asking the professors, is there a possibility to have elephant heart samples?
Yeah.
So you can isolate RNA.
I think there's a tissue sample.
I mean, elephant.
Small tissues.
Tissue. When they naturally die, you can work with the forest officers, Yes. I mean, naturally dying elephant, you can collect the heart, right?
Yeah.
I mean, you preserve the samples, everything.
Yes,
You can take some elephant, naturally die. You are not asking any elephants or you are killing it, When they naturally die. Yeah, you have to set everything and wait for one day, you get one sample, then you get plenty of data. Yeah.
Just one more small question. So about this alpha to beta in human, the switch occurs in the very beginning during the developmental process. So by chance, can we control the switch? So like suppose we know that, okay.
You can control the switch at the transcription level.
Yes.
Yeah.
Like suppose C protein has a lot of mutation and that is causing a lot of problem. So can we switch the expression of alpha to beta when it is in adult form by some transcription factor, and that can accommodate those faulty C protein?
Yeah.
I mean, is it,
Yes,
can it be done?
so for example, the technology called CRISPR-A, the new technique.
Okay.
This CRISPR-A can go and bind to the promoter of interest, activate the transcription.
Okay.
So
Like-
it is possible.
Like suppose like there is a competition between the peptide you inserted, and then like suppose alpha is also equally expressing. Even the rate will be higher, but at least there will be something to compete with or
100%.
complement with.
Yeah, there is a possibility.
Okay.
Yeah. Good question. There is one, the last. He's not a student, but.
Sir, after mutation, how you overcome the natural repair mechanism? After inducing the mutation in mice, in particular peptide, how you overcome the natural repair mechanism?
How do you overcome the natural? So again, treatments is mostly for the improvement or try to rescue as much as can. If you can improve a quality of life of a patient, 1 year, 5 year, 10 year, anything today is better, right? So you are working towards that kind of goal, but not completely reverse, because you still have the mutation. You born with the mutation. You have the mutation. And so either you have to correct it completely at one early stage or before birth or after birth in a quick time. Otherwise, you have the mutation forever, so you have to have a permanent treatment option once you have the phenotype. Yeah. Questions from master students? Not from you. You can ask. Yes, anybody in the back seat? Otherwise, I am going to pick up your name. I have all the names here.
So you said in humans, before birth we have alpha myosin, after birth we have beta myosin.
Correct.
So the genes that express alpha myosin must be switched off after the birth through some kind of mechanism like decapping or... is there any mutation in which that mechanism gets turned off and both alpha and beta myosin express simultaneously?
I don't know, it is possible. If the transcription factor failed to switch, right, then there is a possibility, but I don't know any literature top of my head.
Sir, one more question. You showed the pictures of animal models that are used for study of the disease. You showed the picture of a chimpanzee, which is a very higher order primate.
Yeah.
Is that actually used in the study?
Yeah, they used for hypertension, some natural, cardiovascular diseases. So in Germany, in some places they have the primate facility. They study some cardiovascular diseases.
Because they are endangered animals and also quite big, and they can pose threat to the humans. So
I don't know how they maintain that, but possible. Okay, let's have a tea break and then we will meet in 10 minutes.
Thank you so much, sir. This was really an interesting talk, the way you explained with all the pictures and videos. And the most interesting part which I liked and all the audiences is the organoid structure part. That is something very new.
Thank you.
So yes, definitely we are looking forward to learn more from you. And yes, we will go for a coffee break and we will meet after 15 minutes. Thank you.